Hi, my name is Chloe. Could you please help me with this question? Thank you.
Chloe, this is the wild card question because it wasn't on evolution or plant hormones. So let's have a look here. What have we got? Okay. The diagram below represents what occurs. Let me just change my pen color here. No, let's go with, let's do red. Okay, so this happens during protein synthesis. So if we look at protein synthesis, the word protein, we all know, it's made up of a whole bunch of amino acids. Synthesis means to make. So the word protein synthesis tells you we're going to be making proteins. All right, so first thing you do when you get a diagram is you label it. So for A, that's going to be a ribosome. Okay, B, that's our messenger RNA. So it's a little m and capital RNA. Then we look at these guys. Oh wow, this little guy that looks like a little cross, that is our tRNA. Oh, something else, your messenger RNA is a codon. It's a little triplet base. And your tRNA is an anticodon. All right. What is D? D is what does transfer RNA carry? It carries little amino acids. All right? And E is that bond there. So it's the bond between amino acids, which means it is a peptide bond. All right, let's look at our questions. The first one says identify the process above. So the first thing is that the process is translation. All right? And people, you've got two processes that happen during protein synthesis. The first process happens inside the nucleus of the cell. And that is when we have translation. So just listen carefully. Translation is easy because what happens during translation is that the double helix DNA unwinds and it starts to unzip as those weak hydrogen bonds break between the two strands. And that's caused by an enzyme. Remember, it's caused by an enzyme called RNA polymerase. And that will cause the unzipping. Now you've got these two strands are open. It forms a fork. So what happens is the DNA literally attracts. You've got one strand will be a template strand, and it will attract free nucleotides that are floating around in the nucleoplasm. And those nucleotides will form a complementary triplet base to what is coded on that DNA. And that becomes your messenger RNA codon. All right, so that is transcription. You've transcribed the message or the recipe from the DNA to the messenger RNA. Remember, DNA can't leave the nucleus. So the messenger RNA gets ready and zips through the nuclear pore into here, into the cytoplasm. But now, it's all fair and well because your ribosome is like a factory. This is our protein-making factory in the cytoplasm. So the messenger RNA cruises along and it gets to the ribosome. And it says there, it's now bringing the recipe. I'm going to put this in inverted commas. The recipe, which it got from the DNA. So during transcription, DNA makes RNA. During translation, that recipe that you're getting from the DNA, so during translation, the RNA makes the protein. So if you remember that, transcription, DNA makes RNA, those free RNA nucleotides that are floating around, and then it codes for the messenger RNA, and during translation, which is what's happening here. So this is translation. Here we are translating what the messenger RNA codon is bringing from the DNA. Because the DNA's got the recipe. Alrighty, let's go back to our questions. So it's translation. Please learn. They will ask translation or transcription. You will get it. It will be in the exam. Learn it. It will be in your prelims and it will be in your finals. Alrighty, now, so it's translation, as simple as that. And remember, with translation, here the RNA makes proteins. All right? And I'm going to put here, I'm going to put it in a little block here so you don't forget. Transcription, the DNA makes RNA. And the RNA that comes out of there is going to be messenger RNA. And messenger RNA is your code on. All right, like your transfer RNA is your anticodon. So it says identify organelle A. So we've already done that, so we just go up. A is the ribosome. That's our little factory. And molecule B is our messenger RNA. And the reason the M is written with a little letter is because it's just telling you what this RNA is. Remember, we've got 3 types of RNA. We've got messenger RNA, which brings messages from the DNA to the ribosome. We've got transfer RNA, which its main function, because of its shape, it picks up specific amino acids. And it will then pick up the amino acid that it's told to pick up, or it understands to pick up from the codon of the messenger RNA. Okay, so that's your transfer RNA. And then you have your ribosomal RNA, which literally punts that amino acid that it got from the transfer RNA, make sure that it joins to the next one with a peptide bond, and out it goes as a polypeptide chain. Polypeptide chains all bond together to form different types of proteins. Okay, and then the bonded E, I'm not even going to go up and look, there's only one bond. It's a peptide bond. All right, now let's see. Give only the letter of the molecule that. Now people, you see here, only and letter. I can't tell you how many times I have marked exam papers and I've had learners, you are asked, only provide the letter for, or give the letter for, and they give the name of the thing, or they give the label name, or they start describing a process. You get naught. They're not asking for anything. They're asking for the letter, nothing else. So let's have a look at our letters. It says, what carries the amino acid? Now we know the amino acids, I've just told you, are carried by the transfer RNA, and transfer RNA was C. So you do not write transfer RNA. They say only the letter, so that would be C. Is copied from the DNA? We know it's copied from the DNA. It's the messenger RNA, the codon, and that was B. So, yay. And then the monomer or building block of proteins, we know is amino acids, but what is the letter for amino acids? It's going to be that one, which is D. All right, now I've still got some time here, so I want to do this with you. I want to show you this. Okay, we have DNA, we have RNA. Okay, we have purines and we have pyrimidines. Okay, let's just go down up a bit. And purines. Those are the nitrogenous bases that we have on our nucleotides. For DNA, we have adenine and guanine, and our pyrimidines are, I'm going to write it out, thymine and cytosine. Now look here. You see the Y, and the Y. So that's how you'll remember pyrimidines have got the Y in the word. So thymine and cytosine and adenine and guanine. Now, adenine always complements thymine, and guanine always complements cytosine. So remember, A to T and G to C, always. And write that at the top of your page. If you have any question on protein synthesis, any question on DNA, on nucleic acids, you write ATGC, so you never, ever, ever get confused. Now, if I look at RNA, RNA also has the purines adenine and guanine, but it has cytosine too. But instead of thymine, RNAs will always have uracil. So think of the uracil as a Y with a short leg, the leg's cut off. So your pyrimidines, thymine, cytosine, and uracil. RNA, if you see anything, if it says CAT, we know that it's DNA. If it says CAU, it is going to be an RNA molecule, whether it's transfer, messenger, or a ribosomal RNA. If it's got the U, then it's an RNA. Okay, so people, if you remember that, you cannot go wrong. And every 3 nucleotides, so every triplet base, is going to give you an amino acid. So an amino acid will always be made up of 3, and there are 20 known amino acids to humans, to man, to mankind, to womankind, to peoplekind, okay? 20. And if you think of the combinations, you end up with these 3, with those 4 letters, with an A, a G, a C, and a U for your codons. Well, work it out. You can only ever get a total of 64.